Now we had seen about the first form of cell death, which is necrosis. Next we'll move on to the second type of cell death, which is apoptosis. So unlike necrosis, apoptosis is going to be both physiological and pathological. So physiologically, where do you see apoptosis? During organogenesis. So we don't have all the embryogenic rests in the body, right? So whatever organs have been developed, the embryonic rests are going to be lost. Even thymic tissue is being lost in adulthood, right? So that is all going to happen because of apoptosis. Other than that, hormone dependent tissue can also be involuted. So post pregnancy, what happens? The gravid uterus decreases in size and goes back to its normal size, right? So there again, this happens because of apoptosis. Pathological examples of apoptosis will be usually misfolded proteins. So any disease in which misfolded proteins are implicated, so the misfolded proteins are going to be accumulated, and that will result in the form of cell death called as apoptosis. So remember, whenever misfolded proteins are there, it will the cell will die by apoptosis. So what are the examples of diseases in which misfolded proteins are there? Alpha 1 antitrypsin deficiency. So it is going to cause panacenar type of emphysema along with liver disease. So alpha 1 antitrypsin deficiency. Other than that, neurodegenerative diseases like Alzheimer's disease. Then you also have Prions disease. So all of these diseases can result in misfolded proteins, resulting in the cell death called as apoptosis. Also in viral hepatitis, we can see the apoptosis. So the best example is seen in the hepatitis C virus infection. So this image is actually representing a councilman body, which is also called as a apoptotic body. Okay, so councilman bodies are apoptotic bodies. So these are all the hepatocytes, normal hepatocytes. But look at this cell. It is kind of small and it is having a densely eosinophilic cytoplasm and a very clumped kind of a nucleus. So this kind of cellular shrinkage along with nuclear chromatin condensation, which is pycnosis basically, is the classical morphology of a apoptotic cell. Okay. So in necrosis, we saw it was cellular swelling associated with pycnosis, karyorrhexis and karyolysis. But in apoptosis, it is going to be cellular shrinkage associated with nuclear chromatin condensation alone. Okay. So in viral hepatitis, you see this councilman bodies. So here in apoptosis, unlike necrosis, there is not going to be any membrane damage. So membrane damage is not going to be there. So when membrane damage is not there, obviously there is not going to be any inflammation, right? So membrane is going to be intact in apoptosis. It is an active process, and here it is going to be a caspase dependent programmed cell death. So programmed cell death in the sense there is going to be set pathways and regulatory genes which are going to regulate this kind of cell death. So it is one of the forms of programmed cell death. So there are other forms of programmed cell death also. So remember, apoptosis is one form of programmed cell death which is going to be caspase dependent. And this caspase are enzymes actually. They have a cysteine residue in its active site, so it is given the name C. So C is for cysteine, asp is for aspartate, and ACE is for the enzyme. So this is the full form of caspase. So caspases are going to be activated in apoptosis, and the caspase activation is going to be the hallmark of apoptosis. So hallmark of apoptosis is going to be caspase activation. So the type of caspases which are activated in apoptosis is different. So we have two types of caspases. Initially we have an initiation pathway, then an execution pathway. So under initiation, you are going to have an intrinsic or a mitochondrial pathway and an extrinsic or a death receptor pathway. So all of these intrinsic and extrinsic pathways will result in the activation of the initiation caspases. This initiation caspases will in turn activate the execution caspases. So execution caspases finally lead to the apoptosis. So let's see the various caspases. So the initiator caspases are caspases 8, 9 and 10. Out of this, caspases 8 and 10 are going to be there in the extrinsic pathway. Extrinsic pathway was the death receptor pathway. So in this 8 and 10 are present, while caspase 9 is going to be activated in the intrinsic pathway. So both of 8, 9 and 10 are the initiator caspases. So remember 8 and 10. So 8 starts with the letter E, right? And 10 is going to be represented by the letter Roman letter 10 X like this. So it resembles X, right? So remember EX for EX. So this is how I remember it. So extrinsic pathway, it is going to be caspases 8 and 10, while intrinsic pathway it is caspase 9. Then these caspases 8, 9 and 10 are going to activate the executor caspases, which are caspases 3, 6 and 7, out of which caspase 3 is going to be the most important caspase. So in apoptosis, I mentioned caspase activation is the hallmark, while in neuronal apoptosis, in the brain we don't have caspase. So the hallmark of neuronal apoptosis is going to be activation of a substance called as apoptosis inducing factor. Apoptosis inducing factor is going to be activated in neuronal apoptosis. So I mentioned apoptosis is a programmed cell death, meaning there are going to be certain genes which are going to regulate it. So let us see about the genes which regulate this apoptosis. We have pro-apoptotic genes which promote apoptosis, then anti-apoptotic genes which prevent apoptosis. Then we have stress sensors which actually trigger the cell injury. So it is the one which senses that the cell has been injured and it will start to promote the apoptosis. Okay, so normally there is going to be a balance between this pro-apoptosis and anti-apoptotic genes, but when there is a cell injury, what happens? This anti-apoptotic genes will be decreased and pro-apoptotic genes will be elevated. So it will result in the apoptosis. So let us see what are the pro-apoptotic and anti-apoptotic genes. So starting with the pro-apoptotic genes, the most important one is the BAX and BCL excess. So these along with P53 and even glucocorticoids stimulate apoptosis. Okay, so BAX and BCL excess are the pro-apoptotic genes, while anti-apoptotic genes are BCL2, BCL-XL and MCL1. Then here sex steroids prevent apoptosis. And the stress sensors are usually called as BH3 only proteins, and under these BH3 only proteins we have BIM, BID, BAD, PUMA and NOXA. So you'll have to remember these. So remember it like this. So BAC and BAX resemble like BAC only, right? And all the BCL related things are usually anti-apoptotic except for BCL excess, which is a pro-apoptotic gene. So other BCLs are BCL2 and BCL-XL, which are anti-apoptotic. And other than that, anti-apoptotic, again you are going to have another CL only, which is MCL1. So BCL2 is the most important anti-apoptotic gene, and in certain tumors or lymphomas, this BCL2 is going to be inactivated. So when this is going to be inactivated, apoptosis cannot happen. So the cells will live for a very long time. So one such example is a follicular lymphoma. So a translocation involving this BCL2 is going to be there, which is going to result in the decreased apoptosis, or so the cells become immortal basically. So that is seen in follicular lymphoma. Then other than that, this MCL1 gene, so this MCL1 protein is also responsible for chemoresistance in tumors. So some of the tumors tend to express this anti-apoptotic MCL1, and that is how they have this resistance to the chemotherapy. So upregulation of MCL1 can be seen in certain tumors, which can contribute to the chemo resistance. So in chemotherapy, when you give chemotherapy, the tumor cells die. So what kind of cell death is happening actually? So there is apoptosis and necrosis happening, but it is more of apoptosis. Okay, both apoptosis and necrosis happen during chemotherapy, but more of apoptosis. So next we'll start with the mechanism of apoptosis. So mechanism of apoptosis is going to happen in two phases, which is the initiation and the execution phase. Okay. So under initiation phase, we have two pathways, basically the intrinsic pathway, which is also called as the mitochondrial pathway, and an extrinsic pathway, which will be a death receptor pathway. So both of these pathways are going to initiate some caspases, which will then activate the execution phase. So let's start with the extrinsic or the death receptor pathway in the initiation phase. So for this, as the name suggests, there is going to be some death receptors. So what are the death receptors? They are going to belong to the family of TNF. So TNF family, there is going to be this CD95 or FAS, which also belongs to this TNF family. So these are death receptors present on the surface of the cell membrane. So when the T cell or some other cell which has this ligand to this death receptor CD95, that is, these T cells or the cells which have the ligands will be FAS ligand or CD95 ligand. When these come and attach to the CD95 cell here, CD95 receptor over the cell surface, it is going to cause activation of this CD95 receptor. So it will start forming a trimerization of this receptor. So a single CD95 will be attached, then it will recruit more CD95 which is going to be attached to the phosphligand. So this is called as trimerization of the receptor. So trimerization will take place, and this in turn will recruit a protein called as FAD. So FAD protein is the one which is responsible for the conversion of caspase 8 to procaspase 8 to caspase 8. So this is the active form. So FAD is going to activate the procaspase 8 into active caspase 8. So active caspase 8 is the extrinsic caspase which is being activated. Okay, so this is the final stage of extrinsic pathway. So there is a protein called as FLIP, which is going to be a inhibitor of this extrinsic pathway. So this FLIP is going to inhibit the conversion of this procaspase 8 to active caspase 8. So what is the inhibitor of extrinsic pathway? It is going to be FLIP protein. Then we'll see about the intrinsic or the mitochondrial pathway in the initiation phase. So for this, suppose this is the cell I have drawn in this box, okay. So there is going to be an injury to this cell. So whenever there is an injury, it is going to be detected by the BH3 only proteins. So which were BIM, BED, BAD, PUMA and NOXA. So these BH3 only proteins are going to sense this injury, and they will change the balance between the apoptotic and pro-apoptotic genes, and they will favor a pro-apoptotic gene. So pro-apoptotic genes were BAC and BAX, right? So this BAC and BAX will be activated on the mitochondrial membrane. So what will happen? This BAC and BAX are going to form some oligomeric channels, and they'll insert themselves on this mitochondrial membrane like this. So they are going to form these channels. So what happens because of the formation of these channels? There will be certain proteins present inside the mitochondria which are going to be leaked out into the cytoplasm. So the cytochrome C is one such protein which was present inside the mitochondria. It has come out into the cytoplasm now. This cytochrome C is now going to attach with the protein in the cytoplasm called as APAF1. So APAF1 is apoptosis activating factor 1. So the cytochrome C plus APAF1 is going to form a complex called as apoptosome. So this apoptosome is the complex which is responsible for the conversion of procaspase 9 into active caspase 9. So active caspase 9 is the final step in the intrinsic or the mitochondrial pathway. So there is also some other protein called as inhibitor of apoptosis. So IAP is inhibitor of apoptosis, which is present in the cytoplasm normally. So this, its normal function is to inhibit this apoptosome. Okay. So whenever this BAC and BAX was activated and they form this oligomer channels, I told cytochrome C is going to come into the cytoplasm, leak into the cytoplasm, right? So along with cytochrome C, there is another protein called as SMAC diablo, which is also going to leak into the cytoplasm. So this SMAC and diablo in turn will inhibit the IAP or the inhibitor of apoptosis. So since SMAC and diablo is inhibiting this inhibitor of apoptosis, this inhibitor of apoptosis will not be available to inhibit the apoptosome. So as such, what is happening? SMAC and diablo is going to be a pro-apoptotic factor. Okay. So don't confuse. SMAC and diablo will inhibit inhibitor of apoptosis, and so it is a pro-apoptotic factor. Now that initiation phase is complete. So in the extrinsic pathway, we had caspase 8 which was activated in humans, and then we'll have caspase 9 which was activated in the intrinsic pathway. So these activated caspases 8 and 9 are going to activate the execution caspases which are caspase 3, 6 and 7. So when these caspases 3, 6 and 7 are activated, they are going to activate the certain enzymes like protease and endonuclease and phospholipases. So this endonuclease is going to act on the nucleus and cause internucleosomal cleavage. So the nucleosomal cleavage is going to happen, and it will happen between 180 to 200 base pairs. So for every 180 to 200 base pairs, it is going to cleave the nucleosome. So this internucleosomal cleavage will result in the will actually cause the nuclear chromatin condensation. And this nuclear chromatin condensation is called as the characteristic of apoptosis. So the hallmark of apoptosis was caspase activation, while characteristic of apoptosis is nuclear chromatin condensation. So nuclear chromatin condensation is nothing but pycnosis only. In necrosis we had pycnosis, karyorrhexis and karyolysis. In apoptosis it is going to be just nuclear chromatin condensation. So once this enzymes like protease endonuclease are being activated, one part it is going to cause this internucleosomal cleavage, and other than that it is going to form the apoptotic bodies. There is going to be formation of apoptotic bodies as a result of the activation of this execution caspases. So what are these apoptotic bodies? These are nothing but membrane bound organelles only, which may or may not have a nucleus. Okay. So the part of the cells organelles are going to be disintegrated now. So they'll take a part of this cell membrane and they'll form membrane bound organelles, which are going to have either a part of the nucleus. They may be present, nuclear parts may be present or may not be present. So this is the definition of apoptotic body. And this apoptotic body, what happens is, since it is having the membrane, right? So the membrane which was derived from plasma membrane only. So normally in the plasma membrane, if you see, there is a phospholipid called as phosphatidylserine. So this phosphatidylserine is usually present on the inner leaflet. So we all know the plasma membrane which is going to be tri-layered, right? So in the inner membrane only we are going to have this phosphatidylserine normally. But in apoptotic body there is going to be flipping of this phosphatidylserine, and this phosphatidylserine comes to the outer surface. Okay. Why does this happen? This flipping of the phosphatidylserine will actually ensure that the macrophages will recognize these phosphatidylserine and they eat up this apoptotic body. So macrophages will eat up the apoptotic body, and which is given a special name called as efferocytosis. Okay. Efferocytosis is nothing but macrophages phagocytosing this apoptotic body. And this efferocytosis will happen just like that. So very soon it happens that normally in a microscopic slide, if you see microscopically, you will not be able to identify apoptosis as much as you appreciate a necrosis. So it is it happens very quickly. That is what I mean. Okay. So there is something called as eat me signal. So this phosphatidylserine was one such signal. So this apoptotic body is going to express some eat me signals like phosphatidylserine, calreticulin or C1q. So all of these are going to invite the macrophages. They are going to tempt the macrophages to eat them. So the apoptotic cell is going to express these eat me signals, and the phagocytic cell is going to have receptors for this eat me signals, and when this comes into contact, the phagocytosis of this apoptotic cell will happen, which is efferocytosis. Other than that, there are also find me signals, which are actually recruiting signals. So the apoptotic body is going to express this phosphatidylcholine, fractalkyne, sphingosine 1, ATP or UTP. So all of these are going to be called as find me signals, which actually recruit the macrophages to the site of site where this apoptosis is happening. Then there is also do not eat me signals, that is like normal cells. So normal cells, we don't want apoptosis to happen, right? So the normal cells will express this CD47 and CD31. So when phagocytes which have the receptor, they will recognize this as a normal cell and they'll leave them. Okay. So these are do not eat me signals. So tumor cells also use these signals and escape from apoptosis. So one of the mechanisms by which tumor cells escape from dying is evading the apoptosis, right? So that is how this is, because they express these do not eat me signals, which is CD47 and CD31, and they escape from apoptosis. Next we see about the how to detect apoptosis. Usually on H&E stain, I told it is difficult to recognize apoptosis, but still on H&E stain, that if you remember, we saw the Councilman body, which is also an apoptotic body, right? So how was the cell? It was showing cell shrinkage with increased eosinophilia of the cytoplasm. So there is dense eosinophilic cytoplasm in these cells. Along with that, there is going to be nuclear chromatin condensation, so which was pycnosis. So this is how an apoptotic cell will look on a microscopy. So what is the marker of apoptosis? So we have a stain called as annexin V. So annexin V stain can be used for detecting this apoptosis. This actually detects the flipping of phosphatidylserine. So the flipping of the phosphatidylserine will be detected by this stain, and that is how you find out that this cell is undergoing apoptosis. So what is the molecular marker of apoptosis? So as the name suggests, there is going to be some molecular study to identify this marker. So what is that? It is going to be the CD95. So CD95 or the FAS, we can identify with flow cytometry, and this is going to be the molecular marker for apoptosis. Other than that, we can use certain staining techniques like tunnel staining, DAPI staining, and propydium iodide staining also for detecting apoptosis. Then you can use FRET, which uses some fluorophores to identify the apoptosis. Then most importantly, you can use gel electrophoresis. So on gel electrophoresis, if you see, this is a normal control. So this first image is the normal control. So normal cell, if it undergoes gel electrophoresis, it will look like this. But if a cell is showing this kind of a step ladder pattern, so if you see, there is going to be some breaks in between. So the step ladder pattern of electrophoresis, gel electrophoresis pattern is seen usually in apoptosis. Okay, and it is going to be because of the internucleosomal cleavage. So internucleosomal cleavage, which we saw in apoptosis, was the one which is responsible for this step ladder pattern. Okay. But remember, this step ladder pattern is not specific for apoptosis. It can be seen in necrosis as well, because internucleosomal cleavage, which led to the nuclear chromatin condensation. So this nuclear chromatin condensation can also be seen in necrosis, right? So step ladder pattern is not specific for apoptosis, though seen more in apoptosis, it can also be seen in necrosis. The third image, if you see, it is looking like a tongue-shaped peripheral smear, right? So this kind of smearing pattern is going to be seen in necrosis. Okay.